Welcome to the Science for the Public lecture series. Science for the Public is an organization committed to bringing science information and issues to the general public. Visit our website for our program listings and blog.
The study of viruses is one of those fields that's in its heyday, and we are honored to have as our guest tonight someone who's working on the very frontier of virus research. John Connor is an assistant professor of microbiology at Boston University School of Medicine. He received his PhD from Duke University and joined the faculty of Boston University in 2006. His background and interests are well suited for the kind of overlapping fields that we see increasingly. In his case, genetics, molecular biology, and other areas of expertise. Dr. Connor's virus research focuses, as he says, on understanding how a virus accomplishes the hostile takeover of a host cell. He uses a particular prototype virus, which he'll describe, and he investigates the mechanisms of this virus attack. But in addition to unraveling these very complex processes, Dr. Connor's research is geared toward the development of potential antiviral drugs and vaccines. We are very pleased to welcome Dr. John Connor.
Thank you. I'm very excited to be given the opportunity to talk to you guys tonight, to hopefully tell you with some clarity about the stuff that we do in my laboratory and to convey to you guys some of the excitement that I feel about the work that we're doing and the progress that we're making. I gave the talk the title of Unraveling the Secrets of Viruses. I promise I won't try to tell too many. It's not that we know all of them. But I wanted to really sort of start out by sort of giving you an idea of my background. And I'm a microbiologist. You can see Gary Larson's interpretation of me right here, studying for my midterm. And in general, being a microbiologist means I study really small things. And in my case, I study viruses. And I wanted to spend a moment distinguishing viruses from bacteria. They're often conflated in the media. They both cause us to get sick, and it's easy to confuse them because nobody can really see them without lots of magnification. So bacteria are nasty bugs. They can replicate and divide on their own. They can live in environments, so you could pick up something from the soil, for instance. And genetically, bacteria are incredibly complex. They have thousands of genes. In contrast, viruses are, on the one hand, much simpler. They can't live on their own. They can't make copies of themselves without help, which means they have to find a host. And that means they then have to steal things from the host. And that's sort of the central aspect of what I'm going to talk about, is understanding what they steal and really how they go about stealing it. And what I want to drive home at the beginning is that viruses are very dangerous. They're a current and past threat to public health, to human health, to animal health. The most sort of incredible examples are that of smallpox, which is responsible for at least 300 million deaths in the 20th century. Thankfully, it has now been eradicated. But there's also the example of 1918 Spanish flu, which was responsible for at least 100 million deaths. HIV is currently an endemic problem. And these are just essentially the large hitting infectious disease stories from a virus standpoint. There are also additional stories that most of you have probably heard of. There are a lot of emerging viruses, from H1N1 flu, which we heard a lot about over the last couple of years, to the SARS coronavirus, to Ebola and Marburg, the Lassa hemorrhagic fever virus. And all of these viruses are things that are really, they come out, they hit the public in various different ways, from the scare that surrounded the SARS response to the real danger and worry that surrounds potential hemorrhagic fever virus outbreaks. Here you can see in the picture over to the right, this is a team going out to try to identify whether there is in fact an Ebola outbreak. You can see that they're all suited up. They have tried to cover every part of their body, and they're doing their very best to keep from being infected. So these viruses are incredibly dangerous, and because of that, I want to try and make the case that we need to know a lot more about them. For instance, how do they make us sick, and how do we detect them as quickly as possible? And I'm particularly interested in how we might develop new vaccines, new treatments to keep you well, and also how we might limit them so that they can no longer cause disease. And the central aspect of this is, as I sort of alluded to in the first slide, is viruses are really a paradox. They're both very simple, but they're very complex. So they're tiny, they're microscopic, and they're very genetically simple. Many viruses have really small genomes. So these genomes encode for 5 or 15 proteins. So think of a toy boat. And on the other hand, they infect us. We make about 30,000 proteins, maybe more. And so it's the equivalent in the same analogy of being a Nimitz-class aircraft carrier. And it's sort of strange to have the toy boat be able to strongly affect an aircraft carrier. So how is it that something so simple can completely take over something that is so much more genetically complex? And that's really where my study has focused. And what it comes down to is that viruses follow a very set and simple pattern that turns out to be very damaging to us. Simply put, viruses, when they start their infectious process, they attach to a cell. They then inject their nucleic acid, and it can be DNA or RNA, it doesn't really matter. And once they get that nucleic acid into the cell, they start making copies of it. They start making proteins, and they put the proteins and the nucleic acid together, and they have progeny virions, so they have copies of themselves. And what is illustrated on this slide is one single cell, how it happens. But as you can imagine, if a virus gets into me, it doesn't just find an isolated cell, it finds a whole bunch of them. So if you start looking at what happens just a little bit beyond that, when a virus infects a cell, it makes copies of itself. Those copies go into the surrounding cells, and they make copies of themselves. And this sort of exponential growth of this virus is what ends up giving us not the virus infection, but the virus consequence. And that can range from sneezing and coughing, to rashes, to just generally feeling crummy, to in some more severe cases, hospitalization, feeling really bad. And it's these aspects of virus infection that are troublesome and that we want to fix. But they all start from that simple infection of one cell. And so where my research builds from is trying to understand how it is that viruses infect cells. How do they get in? How do they do all the things that they need to do? And we ask this question using a very simple, what I'll call a model virus. Some people call them prototype viruses. And this is a virus that does not commonly infect humans. It's very easily used in a laboratory setting, and it's well studied, so we have a lot of tools for biological application, ways that we can ask questions and get answers really quickly. And the model virus that my lab studies is called Vesicular Stomatitis virus. You may hear me call it VSV. This is just an explanatory slide for VSV. Up in the top left here, you can see an electron micrograph. It looks a little bit like a bullet. What it does in the wild is it will infect cows, it'll infect horses, it will also infect sandflies. There's not an extensive record of VSV infecting humans in any way. And so this is, in the bottom left, what I've drawn out or have really stolen from a website is a cartoon image of what the virus looks like on the protein level. And the virus makes 5 proteins. It makes one protein that sticks in a lipid envelope bilayer, And then it has several proteins that are responsible for wrapping up the genome and making copies of it. And those 5 proteins are all that the virus makes. So its entire genome is illustrated down here. There are 5 genes in the genome that are transcribed into 5 mRNAs that are what is sufficient for making new copies of the virion. And all of this that the virus does, it does while replicating in only one part of the cell, and that is the cell cytoplasm. It manages to get in, get its genetic material in, and do all its replication, and then exit out of the cell without needing any of the cell's DNA or any of its nuclear material. It solely replicates in the cytoplasm. So reasonably simple, and yet still fairly complex. How is it that it does so many things with such a small group of proteins? And one of the reasons we ask these types of questions with the model virus is that I'm at a university, I do teaching, and so I train lots of people from an undergraduate level to graduate level to postdoctoral training, and I'm really committed to doing that, and I'm committed to doing that without infecting people and making them sick. So the strength of using a model virus is there's a very low risk of accidental infection. It's also a very simple genetic system, so we can make changes to it very easily. And one of the things that we found over the years, and I don't mean to say that I have found, but that researchers have found, is that looking at these simple viruses has direct applications to other viruses, viruses that may be more pathogenic, may be less safe to work with in the lab. So things that we can do quickly and readily with a prototype virus can have implications for viruses from influenza to hemorrhagic fever viruses in a very short period of time. So I'm going to tell you 2 stories tonight. The first is going to be the identification of how virus and cell interactions work and how we figure that out. And I'm going to tell you only one part, and that is how VSV, how one virus takes over the protein synthesis apparatus. And the second story I'm going to tell you about is the development of next generation virus detection systems. And this is a story that involves the melding of multiple technologies that have emerged from Boston University to applying to detecting new viruses or even existing viruses. So story number 1 is the takeover of cellular processes. And this starts from the fact that viruses are good at doing some things. They're often found making their own DNA, their own RNA, and their own messenger mRNA. Messenger mRNA is really a blueprint for making proteins. And proteins are the engines or the workhorses of making new virions. However, mRNA has to be decoded. And it is decoded in the cell by a cellular machine called the ribosome. And there is no virus that makes its own ribosome. So all viruses, by definition, have to take over this cellular process. Somehow they have to trick the cell into opening this machine and translating its mRNA into protein for it. And as you can imagine, this is sort of a difficult challenge. And not the least of which is if you consider what happens when a virus enters a cell and makes some of its own mRNA, what it encounters is a cell that already has its own mRNA, and it's chock full of it. So if you have viral mRNA, there's just a little bit of it. So how is it that the viral mRNA somehow gets to the ribosome? And probably a poor analogy is thinking about trying to get on the road at 5 o'clock on Friday on a holiday weekend, and you're 2 miles from the tolls, and you'd really like to get through as fast as you can. How are you going to get rid of all those other cars? What you need is a police officer of some sort, right, to create a lane for you. And it's this sort of understanding of how viruses and hosts compete to get to the ribosome that I have been fascinated by for a couple of years. And so the question is really, can it even do it? And the reason I'm so fascinated by it is that for VSV, for the virus I study, we already kind of know the answer. We know the virus wins, and it does so in an incredibly efficient way. And the way we know that is shown on this slide. What we did in this experiment that I'm showing you is we took cells, we infected them with the virus, and then we labeled newly synthesized proteins. So we can see what's happening at an increasing time after virus infection. We broke open the cells to remove those newly synthesized proteins, and then we separated them on a gel by size, and then we looked at what happened. And so what you can see in this gel, in this lane marked M, these are mock infected cells. You can see sort of a gray smear, and there's some more prominent bands. This is normal cell protein synthesis. The cell is normally making thousands of proteins. After you infect with VSV and look at 2 and 4 and 6 hours post-infection, there's really 2 things that happen that are remarkable. And the first thing that happens is this gray smear disappears. Host protein synthesis totally goes away. But you can see the appearance of these 4 nice dark bands. This is viral protein synthesis. So somehow the virus is doing 2 things. It's blocking host protein synthesis, and it's managing to get its mRNA translated. Now, what I can tell you is the host mRNA doesn't go away. So somehow the virus has developed a way to trick the system. And that's really what I wanted to understand is, how's it doing this? What's really going on? And so to figure this out, we did a lot of protein mutagenesis. And essentially what we did is we looked at most of the viral proteins. I'm going to tell you about one of them, and that's the viral matrix protein. And we've done a lot of work on it. Essentially what we found is that the matrix protein does 2 things. It both blocks host protein synthesis and it promotes viral protein synthesis. And that information seems to be contained in this one protein, but different parts of the same protein appear to control different things. So they're not linked, they're separated. And the way we found that out in one case was by taking the matrix protein and introducing a single amino acid. So this is one amino acid out of 229. What I'm representing it here is this little red slash within the whole matrix protein. And the change was from a methionine to an arginine. And then when you looked at what happened when this mutant protein was put back into the virus, what you found is that one thing changed. So normally what I showed you on the previous slide was that in mock-infected cell, you have this gray smear. And then in wild-type virus infection, that smear disappears and you get your prominent virus bands. When you looked at the mutant, it had changed. Now the prominent virus bands are still there, but now that gray smear has come back. And so this virus is no longer capable of shutting off the host protein synthesis. And essentially the words on the right say the same thing, that now this host protein synthesis continues. There's been a lot of people that have studied this and have come to the conclusion that one of the reasons that the virus normally blocks host protein synthesis is that the host is no dummy. The host, when the virus infects the cell, tries to make new proteins to warn neighboring cells that there's a virus infection, so the neighboring cells can defend themselves. However, if the virus during infection blocks the synthesis of all new proteins, that warning may not go out. So that's one of the things that this protein manages to do. One of the other things that we discovered it did is that it's actively involved in promoting virus protein synthesis. And we discovered this by essentially continuing our mutagenesis, by continuing to knock holes in this matrix protein and asking what happens when we change different parts of it. So what I'd shown you initially was some changes here on the left side of this representation of the protein. When we moved down and made a 4 amino acid change, we saw something that was a little bit different, and that's shown down here on the bottom. In this case, when we infected with the normal wild type virus, you saw, if we look all the way out here on the right-hand side at 8 hours post-infection, you can still see these strong viral bands, and the host protein synthesis has largely disappeared. When you look in the mutant virus, which is denoted by the symbol phi here, you can see that not only is there no host protein synthesis, but the viral protein synthesis has also disappeared. So what it argues is that this protein is really serving two functions. One function is that it's shutting off host protein synthesis. The other is that it is allowing viral mRNAs to be strongly translated. And that can be sort of represented, in my mind, as a traffic cop where when the virus gets into the cell and brings this protein in with it, the cellular mRNA is blocked from getting to the ribosome. Protein synthesis is blocked there. However, it also forms this secondary role of encouraging viral mRNAs to be taken up by the ribosome. So what good is the knowledge? Now that we know how this happens, one of the basic things is we now have the ability to essentially turn the tables on this virus. Instead of having a virus that takes advantage of us, we're beginning to get to the point where we can take advantage of them. We can make viruses that we know are crippled. We have places where we know that the virus is incredibly sensitive. And so we're now applying the knowledge to develop new antiviral drugs by blocking this protein, and also to develop new vaccines and new vaccine platforms based on viruses that have been weakened by including these mutations, by including these changes to the matrix protein that make the virus a little bit weaker. So that's the first story. The second story I wanted to tell you was about developing new and essentially faster ways to detect viruses. And this story actually comes from the existing problem that virus diagnostics can be improved. I'm certain all of us have gone to the doctor at some point and had the doctor say, you have a virus. And it often is much more comforting if you could know what type of virus. And the central problem is twofold. One is clinical symptoms of viral disease is not very specific. If you have the flu, it may present really clearly, but you may have a rhinovirus and it presents like the flu. And treatments for those different viruses are very, very different. And one of the complicating factors is while there are good tests to identify these viruses, they aren't really fast. They can take hours, some of them take days, and it would be really great to know by the time you leave the doctor's office. Just to drive home the point, there are really three main ways of diagnosing viruses right now. There is polymerase chain reaction, or PCR, ELISA, and cell culture. None of them are really something that are going to be right there at an outpatient clinic. All of them take somewhere between an hour and, in the case of virus cell culture, days. And they involve a certain amount of sample preparation, so you can't do it very simply. And while these are very powerful, they're also something that I think can be improved. And the reason I think they can be improved is that I've done a lot of late night research, and in some of my research, I discovered that the future has a very good virus detector. I found this guy, his name I think is Spock, and he was on this TV show, and he has this thing called a tricorder that I then found is going to be invented in the year 2500. And the tricorder is a very good virus diagnostic. You can identify and sequence a virus in about 30 seconds. And this is really what I think we need. So what I'm going to tell you today is how we're trying to work towards that. And I wanted to work toward that explanation by first explaining one of the workhorses that is currently used, and that is the ELISA. And the way an ELISA works is pretty straightforward. What you do is you take a sample and you expect in that sample is a virus somewhere. And in this case, the virus is a little green dot. And so you put in a little microtiter plate well an antibody that's anchored to the bottom of that well, and you wait a little bit for the virus to bind to that antibody. And after that happens, you put in a second antibody, and that also binds to the virus, but this second antibody has some sort of a label on it. It can have an enzyme that makes a color. It can have a fluorophore that glows under a certain type of light. and you'll get an answer. And these are really good. However, all these steps take time. So in the end, you're talking somewhere between 1 hour, 4 hours, sometimes even longer. And so how do you make it faster? And one of the things that I and my colleagues wondered is whether we could detect capture at this middle step, at virus binding to the antibody. Wouldn't that be great if we could just take off the whole right-hand part? So then the question becomes, how do we do it? And the answer is, how do we make, in this diagram, this little turquoise box detect virus binding? So the concept is that if you have something here and there's nothing binding to it, it will be dark. And then if you actually have binding, that somehow you will get that thing lighting up, that it will do something amazing that tells you, ha, something bound to this antibody. So it's a great concept. And we wanted to use something really simple. The idea being what we develop, we want to be able to be ported around anywhere. And so the requirements basically were that we wanted this to be very user-friendly. So it's got to be portable. It's got to be really simple to use. And it's got to use almost no power. It's got to be run off of a battery, like maybe a AA, but hopefully a AAA. And in theory, it has to be able to convert virus detection to something really easy, like me looking at something. And so really what I'm saying to you, all those descriptions boil down to, we need to find a little thing that's a few nanometers long, and I need to be able to see it. So I'm describing a light-driven nanoscale detection device. Great. So where do we find those? And it turns out you can find them at the Boston University Photonics Center. And so a short drive down the road brought me to talk to these guys, and I'm actually working with two of them right now. I'm working with Dr. Hatice Altug and Dr. Selim Unlu on two separate platforms, both of which are light-driven detection platforms. And I'm only going to be able to tell you about one of them, so I'm going to tell you about the plasmonic nanoholes. I think largely because I like saying that term. And so I'll tell you a little bit about what we're doing to develop a plasmonic nanohole-based virus reader. And the idea that this is based on is that plasmonic nanoholes are very interesting physical structures. So if you take a very thin microchip that does not allow light through, and you punch little tiny holes in it, so small that light shouldn't be able to get through, something very strange happens. And that is that one wavelength of light gets through. And more particularly interesting about that phenomenon is that light is really sensitive to what's around those nanoholes. So the question we asked was, what happens if we put an antibody right next to those nanoholes? And the idea being, if a virus binds right there, that the increased mass of that virus should change the light as it comes through. It should make the light shift from one wavelength to another. If everything worked really well, it would go from green to red, which would be very easy to read and very easy to interpret. So we decided to try it. And essentially what we did is we made several different sensors on a very small chip, and they're illustrated here on this slide. You can see the reference sensor up here, sort of off to the upper right-hand side of figure A. These are just a set of plasmonic nanoholes where we haven't put anything on them. The ones in the foreground, we've put antibody right next to these nanoholes. And if you take the reference sensor shown here in A, and you look at light being transmitted through, you see this peak right about 700 nanometers, and if you add virus, that peak doesn't change. That's good. If we then look at this detection sensor, when we add virus and look and see what happens, now what we get is that in blue, you can see just what happened When we had antibody, when we add the virus, all of a sudden the wavelength shifts to the right. And that's the red line. And so what we have essentially is the demonstration of that principle. So we can put virus on these chips. These chips will detect that virus binding. What I'm showing you now is wavelength peaks, but this in the end corresponds to different colors, and we can modulate where those are in order to make those colors fit a very easy interpretation scheme. And so this is just an image of the current assay setup. At this point, we have a detector chip. That's the little plasmonic nanohole arrays that I talked about. You can see them down here. There's a quarter to the right, giving you an idea of what the size is. The detector chip is square. It's a little smaller than a quarter. So this is something that you could imagine wandering around with, being very easy to use. The reader that we have right now is right above it. It's a little bit larger. Right now it attaches to a microscope. And we're working on making this better. So at this point, what we can do is we can detect the model virus that I've told you. We can detect it purified. We can detect it in serum. We can detect it in saline. So it's a fairly robust detection system. And we have also validated detection beyond this model virus to show we can detect hemorrhagic fever viruses. We can detect pox viruses. So this appears to be a very generalizable platform, something that we can use very simply, very easily, and something that we hope we can begin to develop into the tricorder 0.1. What we're especially working on now is an easy-to-use graphic interface so that results can be interpreted fairly easily. And there's some debate as to what we should use. In general, the lab favors a small video screen. My daughter, however, has been very vocal in this, and she thinks that emoticons would be really effective. So that's about all I have to tell you, except to acknowledge that when I sit before you and speak, I don't speak about things that I did alone. I have a reasonably large lab that is illustrated over here, if I can get the cursor to move. And I want to acknowledge especially Judy Yen, Claire Marie Falone, Natalia Mamaeva, Dan Roselle, and Aaron Hodges, as well as Ewan Dunn. And we work well beyond my lab. As I told you about the collaborations we did with the Photonics Center, we've had the help of Helen Fawcett, as well as Selim Unlu's lab and Hatice Altuğ's lab. Most of the work I told you about in the last few minutes was done by Ali Yannick. And what I did not have time to talk about today is we have a fairly close collaboration with USAMRIID under the direction of Lisa Hensley. And all of the work we do, we do with the help and support of national funding agencies, which means we do it with your help. And for that, I'm very thankful. I'll end my talk now, and I'll take any questions you guys might have. Thank you. Yes.
This may be a real dumb question, but you talk about holes that are so fine that they can detect different wavelengths.
Yep.
Are you talking about some kind of a material, or are you literally talking about an organism?
So the question was, what do I mean when I say these plasmonic nanoholes? And I'll say first that this is technology that has been developed by Hatice Altuğ, and so she's much better at explaining this. But what I can tell you, and I should probably sit down and tell you, is that the general principle is that was developed a long time ago by a fellow named Beath, was that if you made holes small enough that so say you have visible light of a wavelength of about 500 nanometers. If you made a hole that was 200 nanometers, the light shouldn't be able to fit through that little tiny hole.
I understand that, but how do you make a hole that size?
So the question is, well, now how do you make a hole that's only 200 nanometers?
It's like when we sent a small hole through a
And so the answer to that question is you use a very high-powered electron beam. And that will make really tiny holes. And if you can control it enough, and people have put a lot of effort into this, you can control it so that you can make regular patterns of them. And the thing that's really strange is, even though light should not get through those holes, a wavelength does. And it's usually a very specific wavelength. And to my knowledge, nobody really understands exactly why. What we know is it happens. And the second thing we know is if we change the things that are around those holes, the light that is emitted on the other side of the chip changes. And so we're basically using that phenomenon. Yeah, I think it's pretty cool, personally. If you were to ask me in another, other people have asked me, and my response has been magic. But that usually doesn't satisfy people. Because in the end, they want to know more than just something happens, poof, and it works. You want to know, well, how do we make more of them? Right? How do we make an entire sheet of these things, cut them into 1,000 pieces, and hand them out so that everybody can use them? And usually magic doesn't work very well as an explanation there. So did I answer the question?
Yeah, it's hard to visualize, using something that'll create a hole that'll literally measure wavelengths, but it sounds pretty fascinating.
So far it's been very fun.
You think of crystals or, prisms or other ways of looking at
Yeah,
wavelengths.
of doing
You
it.
guys are literally looking at it by size, which is pretty phenomenal.
So that's one way. Another thing that we've done that, and this is the work with Salim Unlu, works on the idea of surface reflectance, which, have you ever seen a soap bubble and how you kind of see a rainbow? That's surface reflectance. And you can actually make use of that in a very similar way to detect viruses. And what you can do with that technology that you can't do with a plasmonic nanohole technology is you can start getting size and shape. So you can say, there's something stuck right there, and it looks like a bullet. I bet it's VSV. Or you can say there's something stuck right there, and it's really big, and it's round, and it looks a lot like it's a pox virus. And so we've taken a couple of different avenues to try to apply these really cutting-edge engineering principles and physics principles to virus detection. And all of it centers around the idea that what we would really like to do is remove the need to label something. We'd like to be able to take something, put it in your sample, whether it's blood or serum or saliva, and then pull it back out and ask what's there, without having to walk back to a lab or put it in something that takes a lot of care and maintenance. And the real problem with putting labels on something is often it requires enzymes, and enzymes are really delicate. You usually have to keep them cold until just before you want to use them. And as you can imagine, if you are on vacation in Chile or Argentina, you might not want to carry that with you. You might want to just carry a small chip. And so that's what our goal is to have it be a much more simple detection device.
And how close do you think you guys are to a detection device?
So the question is, how close are we to having this working? And I'll give you the response I gave you a year ago. We're about a year away.
Good response.
So
Are you
each,
just like a little smidgen closer?
well, yes and no.
Things are so small you can't measure.
The problem being every time we think we've got it, we don't necessarily have it. And there's a definite lesson to hubris and being overly confident. So when I say a year, I understand that it may not be. But so what we've done right now is we have this system, and we've gotten it to the point where we have a prototype. And we've taken the prototype, and it has worked in our hands. But that's not entirely fair because, of course, we developed it, and it's not like we're going to walk around to where everybody is. The goal is to have it be something that we can send to somebody else and have them work it. So now we've done that. So we've taken the machine and we've sent it off to somebody else, and now they're going to try it and see if they can get it to work. And if they can get it to work, then we're really on the way. If they can't, then we've got some more work to do. And I'd love to be able to tell you that we know the answer one way or the other, because in either case it's progress. Mm-hmm. But right now they're still testing.
Pretty incredible.
So are there more questions? Yes?
I wonder if we could kind of switch from the technology back to sort of the, I guess, molecular biology.
Yeah.
I was fascinated by that matrix protein and how you were able to modify it to interrogate the cellular protein machinery.
Right.
And so I'm not quite sure how big that protein is, but you talked about making modifications along it. How did you decide where to make those modifications?
So,
or did you do the whole thing?
So the question is, tell me more about matrix. How big is it? Why did you mutate the things that you mutated? And how many did you have to go through before you got an answer that you liked? And the answer is several-fold. First, it's not just me that has approached the problem, and it's not just rational thought that has approached the problem. So the first thing that I showed you, which is changing that one amino acid, that was based on findings that the virus, that you could generate mutants of the virus. You could spontaneously recover mutants that contained that amino acid change. And nobody really knew for certain that that was absolutely responsible until a recombinant system was developed that allowed us to generate viruses completely from cDNAs. And once we were able to do that, we were able to specifically test the question of whether that protein and whether that amino acid itself was solely responsible. And so those studies, which were carried out by people before I even entered the virus field, were really sort of seminal studies. And so one way is that you ask the virus to tell you, and you look for a certain phenotype. But the second part of the story is the second area that we mutated. We had a hypothesis that we were testing. And I have to be honest and tell you it was absolutely not what I showed you. We expected that when we made those mutations, that the virus would no longer be able to assemble properly. And that has to do with something else that the matrix protein does. The way it got its name is it sits in essentially a matrix that surrounds the genome and the lipid bilayer and the assembled virion. And so we figured we were going to break that. And we conclusively proved that by making those 4 mutations, we did not break that. But in the process, in looking at the virus and what it did, what we noticed was this very prominent protein synthesis defect. So in that case, we had an idea, we tested it, and then we kept our eyes open when the first hypothesis failed.
That's a fantastic day in the lab.
It's the day right after the postdoc at the time, who was me, gets ready to jump out of the 5th floor window saying, this hypothesis is totally not true and I can prove it. I think there's another question in back.
Yeah. How would you place the virus on the chip if I was on a vacation in Chile?
Right. So you're in Chile and you feel sick. I would hope you'd go to a hospital. And what they would probably do is take blood. Or they would swab your mouth and they would probably put it on.
So it's not like neat.
I think right now I'll say no. There may be engineers who figure out a way to make a very small device where you could incorporate that. You could imagine incorporating that little chip into something that was attached to a swab, or that was attached to a blood drawing device that would take a little tiny pinprick of blood and suck it onto the chip and go from there. And that's a microfluidics question. And there are a lot of people that are really competent to ask that question and people that are doing that work. I'm not yet one of them. Is there anything else? Well, thank you all. So what got me interested actually was college. And I started out college thinking I was going to be an engineer, and I was pretty excited about that, and was pretty much on that path, had decided a little bit to perhaps look into chemistry, and ended up taking a course from a teacher named Amy Vollmer, who was a microbiologist. And Amy really just sort of brought alive the aspect of how intriguing and exciting bacteria and viruses are. And it set a seed, and it's a seed that didn't really germinate until well after I was in graduate school. And at that point, I began to really understand that's kind of what I wanted to look at and what I wanted to study. And so I moved directly to study viruses, to try and figure out what they do. And it's honestly been very fun. So the aspect of the work that I enjoy the most is actively changing. What I used to say was the best part was actually being in lab, doing experiments, looking at the results and designing what to do next. And while that's absolutely my favorite thing, unfortunately I get to do less and less of it. As I've become an assistant professor, as I have more teaching responsibilities, I get in lab less and less. And I think my colleagues will tell you, that's great. I just make a mess when I walk into lab. But what I really enjoy now is I get to work with a huge cadre of incredibly intelligent people. And now the work that I can do and can help get done spans a much broader picture. There's a lot more things that I can do and a lot more things that can get done. And one of the things that I talk about in the lecture is that we can bring together different disciplines, and that's something that I really can't do just myself. It needs a whole lot of people playing the same sandbox. And in my position now, I can help bring people together to play in that sandbox, and that's actually been very fun. Question of what's the greatest challenge, of course, has two levels, and one of the greatest challenges is the viruses, because they are a constantly changing adversary. You have a virus such as influenza, which you might be immune to one strain, but in a couple years, that same virus that we call influenza looks enough different that our immune system doesn't recognize it anymore. And so part of the real challenge is figuring out how to beat that type of adversary. The more direct question of what's most challenging to my laboratory is probably finding secure sources of funding so that we can spend the maximum amount of time doing the work and the minimum amount of time telling people what we plan to do and okaying that they think these are the right things to do. I think my lab has built a fairly good record of finding neat things and making honest advances. And the longer you leave us to it, I think the more we're going to be able to do. And so a lot of times what we're searching for is the freedom and the space to be able to get that done. So the question of why is it that people should know more about what we're doing comes down to the fact, I think, that when you open papers, when you look at what's happened over the past few years, there have been new emerging viruses. There's recently been a flu pandemic, and there are continually new reports of viruses we've never heard of that are coming out. And what's unique about viruses and is not true about bacteria is if you have a bacterial infection and you go to the doctor has something to prescribe for you. They don't even need to know what that bacteria is precisely. They can get a good ballpark guess and give you something that's likely to be effective. For viruses, that's not true at all. Most of us have had the experience where you go to the doctor and the doctor says, I think you have a virus. Go home. Take a couple aspirin. Call me if you feel worse. And there are a lot of people, I'm not the only one, who are working to try to change that situation, to make it so that we understand a great deal more about viruses so that we can stop the new ones that are emerging and so that we can control the ones that have become endemic and circulate in our population. And it's not really an easy process. I would love to say that I can come in on a Tuesday, get started, and we can have a vaccine or an antiviral drug to one or another virus by Friday. Unfortunately, it hasn't worked out that way yet. So I come in, my lab comes in, a lot of people come in every day, and we do our very best to find those new vaccines, to build the new antiviral drugs. But it's a time-consuming process, and there are a lot of things that we don't know about viruses that we need to understand first before we can design the effective drug, before we can make the good vaccine. And that's part of what I and my colleagues are trying to do.